Hi. So I went to a meeting today with like all the fellow TAs that are here on campus that we TA with. And I was able to ask them about different ways that they explain things. And so I just wanted to go through maybe a different way to explain smooth muscle for those that my last explanation didn't make sense to, which is totally fine because not every explanation works for every person because not everybody thinks the same. So hopefully this can help you. It's not just about smooth tissue, so if you're about to close the window out, I'm also going to go through a couple other hints that they talked about. So hopefully those can help you more or less. So yeah, let's just go through this. Let's share my screen. Okay, so the first we're going to go through smooth tissue and cross section. So this is smooth muscle in cross section. And the first thing that they talked about was you have all these little pink dots everywhere. And some of them are empty. They don't have those nucleuses. But when they do have a nucleus, it takes up almost that entire pink area so that you almost can't see the pink where it has a nucleus. So it takes up all of that area. And so if you compare this to cardiac in cross section, you'll notice that while it still does have the nucleus right in the middle, there's still a lot of area on the sides of the cell for it. So that is how one of the TAs can tell the difference is in smooth muscle, the nucleus, when there is one, it takes up almost the entire cell. Whereas in cardiac, when there's a nucleus, there's still a bunch of space on the edges. And opposed to skeletal muscle here, the nucleus is going to be pushed to the side. So hopefully that helps somebody go through how to identify smooth muscle. Remember to put muscle at the end of your words, so it's smooth muscle. It's not just smooth. Skeletal muscle, not just skeletal. Just because smooth, I don't know. We just went over that in our meeting today as well. Yeah, so those are the muscles. The other thing I want to go through is your neurons. So you're going to have like your nervous tissue, so your white and your gray matter, but then you're also going to have your peripheral nerve tissue. So I kind of want to just go through how to tell the difference. We learned this today as well. So this, I guess what we have up right now, this is your white matter. You can tell because you can see this white part around is your myelin sheath, and the blackish blue part in the middle is your axon. And so this is going to be your white matter and cross section. And this could also be called nervous tissue. You also can have your gray matter, and gray matter is where you can see like the somas and the nuclei, which you could also call your nervous tissue. But with peripheral nerve or white peripheral nerve matter, you're going to see this fascicles, almost like we do in skeletal muscle, where it's just like bundles of them. So we see the fascicles, and this is peripheral, whereas with just the white matter and the spinal cord, we don't see those bundles. So that's a tip they gave us if you were going to go to nervous tissue versus peripheral nervous. I'm trying to think what else would be a good hint they went over, but I can't think of anything else on the nerves. If you have any more question on like nervous system, I did do a video on spinal cord, which should be attached. I also have a hint with the connective tissue. So I do have it downloaded. I was right, I wouldn't have it downloaded. Okay, so this one is with fibroblasts versus mast cells. This one I got from a adjunct or a teacher that I work for, and he talked about, so your mast cells are only going to be in loose fibrous connective tissue, whereas your fibroblasts can also be in your dense fibrous connective tissue. So here, this is an example of your dense, regularly arranged connective tissue. And so these like little, I don't know, they kind of look like nucleuses to me, but they're not. These are going to be fibroblasts because they are in that dense, regularly arranged, and mast cells are not in dense connective tissue. Let me see if I can find. So like if you were to get this picture, this pink part, and it is your dense, irregularly arranged fibrous connective tissue, and the little dots in them are your fibroblasts because you don't have mast cells in your dense, irregularly arranged connective tissue. I'm trying to find some other good slide to show you on. I guess I don't have one. Okay, I guess I don't have one. But if you were getting a mast cell, it would be in your loose fibrous connective tissue, and a mast cell is bigger than your fibroblast. So if you had them, you'd be able to compare them. One good tip to tell the difference between hyaline cartilage and elastic cartilage is with elastic cartilage. Let's see if I can find a good one. I should have planned this out, guys. Sorry, I'm just doing this on the fly. Okay, this works. So this is elastic cartilage. Okay, this one's better. In elastic cartilage, you're going to have like these eyeball-looking things, but then you're also going to have these elastic fibers that run around the middle of them. Whereas with hyaline cartilage, good one. It's more of a glassy look. There's not anything running between them. So this is just hyaline, and if it had lines running through the middles, these are your elastic fibers, so it's elastic cartilage. And then lastly, I just wanted to go show you guys a couple pictures. Maybe if I can find one. So this is a slide that not a lot of people get in their practice quizzes. It is one of your vocab words that you need to know, one of the tissues you need to know. If you see this, it's going to be your blood. We don't really have another like picture we use, but if you get this picture and it's like name the tissue, this is blood. That's all they would ask you about it. They wouldn't ask you any structures or anything. Sorry, I'm trying to think of anything that could help you on the test. I guess one thing, so this is your osteon, this whole thing, and then the middle where it's circled A is your central canal. These like lines that like go inwards towards the eye, I mean not your eye, towards the middle, are called your canaliculus, and they remind me of how like the iris in your eye is kind of texturized. So I always think, can I lick my eye? Canaliculi is how I remember it, just because they kind of like give that eyeball idea. These like little black dot structure things. So if they ask you for a structure, they're the lacuna. But inside the lacuna, you have your osteocytes. So they could ask you, name this cell, and then they're asking for the osteocyte. But if they say name the structure, name the space, they want lacuna. And then you have these rings that go around it. I kind of gave that ring shape. I'm not doing a very good tracing them. They are concentric lamellae, and they're the ones that make the ring. So I just remember C concentric, C circle, and that's what they do. Yeah, that is all of the tips that I can think of off the top of my head. If there's anything else you guys want me to go over, I would be more than happy to do so. Or if you guys want a tutoring appointment, I can do those as well. Just feel free to contact me, and good luck on your test.